So today we will discuss the disease Leishmaniasis, in which we will cover what is Leishmaniasis, types of Leishmaniasis, life cycle, and the current drugs available for the treatment of Leishmaniasis. So it is a group of parasitic disease. It is caused by protozoa Leishmaniasis and transmitted by the bite of infected female phlebotomide scent flies. The protozoa Leishmania belong to the family Trypanosomatidae that infect mammalian mononuclear phagocytic cells. It is caused by the wide spectrum of clinical syndrome, and it is transmitted from animal to human and between human, and can be transmitted vertically from mother to fetus and by blood transfusion. Types of Leishmaniasis. There are four types of Leishmaniasis. Leishmaniasis is also called as Kala Azar, and first one is visceral, mucocutaneous, cutaneous, and post-Kala Azar dermal Leishmaniasis. Visceral Leishmaniasis, that is Kala Azar, is caused by the Leishmania donovani, and in this type the parasite is in the bloodstream and can cause serious problems. Then mucocutaneous Leishmaniasis, it is caused by the Leishmania brasiliensis. Then cutaneous Leishmaniasis, it is caused by the mainly three strain: Leishmania major, Leishmania tropica, and Leishmania mexica. Then post-Kala Azar dermal Leishmaniasis, PKDL. In case of visceral Leishmaniasis, a percent of patients treated here develop a skin condition due to the Leishmania infantum. Now we will discuss the life cycle. So here how it is bite by the sand fly, then it causes the attachment and it leads to macrophage and reuptake. Thereby there will be intracellular MST formation, then multiplication followed by lysis, and these generated intermediate further reinvention that leads to again multiplication here. So this is the life of sandfly. So how they do the work. Then current drugs available for the treatment of leishmaniasis. There are different class of leishmaniasis drug that are available for the treatment. First major class is the antimonial, that has the sodium stibogluconate and meglumine antimonate. Here this is the chemical structure, and the mechanism is to bind ribose moiety and form stable complexes with adenine nucleoside, which act as an inhibitor of Leishmania purine transporters. Then diamidine has the pentamidine drug. Only one drug is available that act by interacting with kinetoplast DNA replication and inhibits topoisomerase 2 and interfere with the aerobic glycolysis. Then polyene. Here one of commonly used antibiotic amphotericin B that is also used for the treatment, and it binds with the ergosterol of cell membrane of parasites and forming micropores, and leakage of cellular content will be occur. Then imidazole class has the ketoconazole drug. It is one of antifungal drug, and this is the chemical structure and inhibition. It inhibits the conversion of lanosterol to ergosterol, thereby impairment of membrane function. Then in other types of drugs, there are two drugs, miltefosine and peramomycin. Miltefosine act by induces the DNA fragmentation and apoptosis in the parasites. Peramomycin inhibit the protein synthesis in non-resistant cells by binding to 16S ribosomal RNA. Okay, thank you.